Cellular respiration. Here are our learning objectives for Chapter 7, learning about cellular respiration and what that is. You probably know perhaps a thing or two about respiration and how breathing in and out, breathing in oxygen, breathing out carbon dioxide is part of our respiratory system, which we use our lungs for. But this is a little bit different. This is about what happens at the cellular level. And all cells need to respire. And in this chapter, we're going to dig into why that is. So first, learning objective, why do cells undergo respiration? And this is going to tie together some things that we've been learning about up till now. All right, so in Chapter 2, I told you about carbohydrates and how carbohydrates are used to produce energy. Okay, so here's glucose over here. And then in Chapter 4, we learned that ATP is used to power endergonic reactions because the release of energy from ATP is so powerful that it can make what seems like pedaling uphill like floating downhill, because there's just so much of a release of energy. It's like putting a jet engine on your bicycle so that when you go uphill, it's like you've got the energy to do it. In the last chapter also, I introduced redox reactions where electrons are moved around, where one molecule becomes oxidized when it loses electrons, oxidation is loss, and another molecule becomes reduced when it gains electrons. So in this chapter, when we talk about respiration, we're going to see connections between all of these concepts. So simply put, glucose is fuel to make ATP. We get ATP by taking the energy stored in the bonds of glucose to make the ATP. And this is how that process works. Learning objective 2, what is the chemical reaction for respiration? So here is your generalized overall formula for respiration. We take some glucose, which is C6H12O6. We take some oxygen, and through a lot of very complex steps, a lot of really complicated processes, we convert that to carbon dioxide and water, and we get from that a number of molecules of ATP. But of course, all energy transfers are inefficient, so some of that energy is going to be lost as heat. So we do kind of burn a little bit of energy. It's not a completely efficient process because no process, no energy transfer is completely 100% efficient. Now, this formula is fairly simple, but really there's a lot of complex steps that happen between starting out with glucose and getting to have some ATP. So one does not simply throw a ring into a volcano. That's not what the Lord of the Rings is all about. That is kind of the summary of it, spoiler alert. But really the thing that makes it a great story is knowing all of the details about how the process, getting from the Hobbit land to the volcano and back again. All right, so learning objective 3, we're going to learn about coenzymes and how they are used in respiration. Okay, so what is a coenzyme? A coenzyme is a molecule that it's not really comfortably fitting into any of our other categories of biological molecules. What we'll see when we look at the structure of a coenzyme, a couple of our coenzymes, is that Coenzymes are kind of a pastiche or a mishmash of different biological molecules. Tend to be kind of small, but they are very important. They interact with enzymes to allow them to function. Remember, enzymes function by lowering activation energy. And many of the vitamins that we get in our diet, like the B vitamins, vitamin C, vitamin A, these are coenzymes. And that's why we need them in our diet, to help our bodies to function properly, because we can't synthesize them in our own bodies. And in cellular respiration, coenzymes are important chemical shuttles. In particular, NADH and FADH2 are electron shuttles. They move electrons around. So they start out in the oxidized form without electrons as NAD plus or FAD, and they get reduced when they pick up electrons to form NADH or FADH2. Coenzyme A is another chemical shuttle. It doesn't shuttle electrons, but we're going to see what it does shuttle when we get to talking about the Krebs cycle. So NAD stands for nicotinamide adenine dinucleotide. So here you can see, here's the molecular structure of it. Don't expect you to memorize this, but maybe you recognize that here is an adenine, or at least you recognize here's a 2-ring nitrogenous base. It's a purine attached to a 5-carbon sugar, attached to some phosphate groups, attached to another 5-carbon sugar, attached to another nitrogenous base. This one isn't in DNA or RNA, but this is another type of nitrogenous base called nicotinamide. Not the same thing as nicotine, so don't get the idea that smoking is really good for you, but named by the same, after the same French fellow, Nicot, who, N-I-C-O-T, so different kind of molecule. But NAD plus, as it is abbreviated, is this form here, and then we can absorb some hydrogen atoms that have both electrons and protons. 2 of those electrons and a proton are going to get put onto that nicotinamide portion of the molecule, and then you're also going to have a proton to form this reduced form of nicotinamide. And if this is all just, you're like, what is going on here? I am confused. Remember this. NAD plus is the electron bus taking electrons to electron camp. NAD plus is the electron bus taking the electrons to electron camp. So when the bus is full of electrons, and that's 2, the bus is full. So NADH is a bus full of electrons. It's got 2 electrons with high potential energy as well as a proton, and they're all going to go to electron camp. And we'll tell you about electron camp later on. It comes at the end. FADH2 is another electron bus. It's not as abundant as NADH. We don't talk about it as much, but it's got the same thing going on where it carries electrons too, but those electrons don't have quite as much potential energy as the ones that get onto NAD plus. So NAD plus is the electron bus taking electrons to electron camp. It's an electron shuttling molecule. All right, so for learning objective 4, we're going to see the processes that turn glucose into the energy that we need to produce ATP. And there are 4 stages called glycolysis, pyruvate oxidation, the Krebs cycle, and the electron transport chain. And we're going to go through all of them, and we're going to see what goes into and comes out of each of these processes. What are all of the reactants that go in and the products that come out. Okay, so for aerobic cellular respiration, which is the kind that's most familiar to us, occurring in almost all eukaryotes, we've got 4 stages, and this is the order that they occur in. Glycolysis comes first, then pyruvate oxidation. Remember, oxidation is lost, so pyruvate is going to lose some electrons. The Krebs cycle, and then oxidative phosphorylation, also known as the electron transport chain. Okay, so a lot of what you need to know about this whole process is summarized in this slide. Glycolysis is the first part that happens in the cytosol, in which glucose is broken down to form 2 molecules of pyruvate. In that process, we get a little bit of ATP, but we also get some electrons that are carried by that electron bus, NADH, over to electron camp, which is over here. Pyruvate oxidation turns pyruvate into this molecule called acetyl coenzyme A. That doesn't produce any ATP directly, but it does give us some electrons. That feeds into the Krebs cycle, which is also called the citric acid cycle. Not to try and be confusing, but good to know that there's a couple of different names for it. And that produces a little bit of ATP also. So when you see substrate level phosphorylation, essentially that means a little bit of ATP is produced in that way. Oxidative phosphorylation, this last part, that is where we get the vast majority of our ATP from. This process produces beaucoup ATP, a lot of ATP. And so we're going to kind of walk through these 4 different stages a little bit more slowly and see what it's all about. And you can see these last 3 steps all occur inside the mitochondrion, which is why we say the mitochondrion is the powerhouse of the cell, because it produces so much ATP. Okay. So glycolysis is the first step in the process. Glycolysis means splitting sugar. Glyco, remember like glucose or glycogen or glycoproteins, it all means sugar. And lysis means splitting. The word analysis means through splitting. So splitting of sugar is exactly what this process does. That's not really the end in itself. The end in itself is to produce some ATP. But the way that that's done is by splitting glucose into 2 molecules of a 3-carbon molecule called pyruvate. Because glucose has 6 carbons, pyruvate coming from glucose is 2 3-carbon molecules. 3 carbons times 2. All right, so glycolysis occurs in the cytoplasm, has 2 major phases. It starts off with an energy investment phase, and it finishes with an energy payoff phase. And what you're going to see, if you watch closely, is that oxygen is not consumed. We don't use any oxygen for glycolysis. So glycolysis occurs whether oxygen is present or it's not. And this is a universal process. All organisms have the means to do glycolysis, whether they're tiny little bacteria or dogs or cats or you and me and everything else that's ever lived on Earth. Okay, so this slide summarizes the kind of the going into and coming out of question as to what are the reactants, what are the products. So in the energy investment phase, we start off with glucose, and then we have to spend 2 ATP molecules. That's why it's the energy investment phase. We have to give glucose a higher free energy so that we can get that energy back. Get some higher energy molecules when we do the actual split of glucose. But when we do, we get 4 ATP back. So you pay down 2, you get back 4. So if we make an analogy to money, it's like we're doubling down, right? I give you $2 today, you give me $4 tomorrow. I'm happy with that arrangement. We also get 2 molecules of NADH. So we're harvesting some electrons from glucose and putting them on the electron bus and sending them to electron camp. So we're also producing some pyruvate, is the final product, plus some water. So here is our net, what goes in on the left, what comes out on the right. Glucose goes in, ATP, or pyruvate comes out, plus water. ATP is put in, but you produce 4, so you net 2 ATP. You pay in 2 NAD+, 2 empty buses. We put electrons on them, and then we send those electrons on the electron bus to electron camp. We get NADH plus 2 protons. But really what's going on is a 10-step process. Each of these steps is catalyzed by a different enzyme. I'm just showing this to you so that you know it's not a simple process. If you were a scientist, if you were taking biochemistry, you might be made to memorize all of this, which I'm not expecting you to. I'm just showing it to you for your own edification, to know that it happens this way, where you pay in the ATP in different steps. And after the energy investment phase is done, you end up with these 2,3 carbon molecules. And those 2,3 carbon molecules are what really go into producing the ATP and harvesting electrons. That all happens in the energy payoff phase. So we get 2 ATP here, 2 ATP here for a total of 4, and 2 molecules of pyruvate. And all of these things you can see are being shown as happening twice, because we've split that 1 molecule of glucose into 2 3-carbon molecules. But this is really what you need to know of it all. This is really the most important part here, especially down here with the what goes in and the what comes out. So at the end of glycolysis, 2 pyruvate molecules, 2 net ATP, 2 NADH, taking the electrons to electron camp. And in the next step, the pyruvate moves on to the exclusive mitochondrion club.